So without further ado, let me introduce the speaker for today. We're really happy to have Dr. Brett Carter here with us. He received his Bachelor of Arts in Biochemistry from Augustana College in Illinois. He earned his PhD in Neuroscience from the University of Michigan, Ann Arbor, and did his postdoctoral work at the Whitehead Institute for Biomedical Research in Cambridge, Massachusetts. Prior to his work here on campus, he was a visiting assistant professor at Oberlin College in Ohio. So thank you for joining us, and the forum is all yours.
All right, thank you, Catherine. Thank you for the kind introduction. And thank you all for coming. I was telling Catherine before this, I was like, I haven't been to talk in over 2 years. So this is just like getting back to the way we do things and share knowledge and experience. And I'm excited to share some of the work that's being done by my students and I in these strange times. So the title of my talk is Exploring the Impact of Environmental Factors on Brain Formation Using Zebrafish. Hopefully it was trying to entice some of you to come or at least watch. And my main goals for today are a fewfold. One is to introduce myself to those who are not familiar with my work and my team on campus, to let you know of what we're doing and how we do things for potential collaborations or ideas that people will be interested in. And of course, to share the exciting findings that we've been making in this area. So I always start with the end, which is acknowledgements, because I feel like that's central to what the progress that we've made. So this is the people I need to thank for providing support for this work in various forms. So the biology department is a fantastic group and unit, particularly my colleagues in molecular biology research suite to have helped with some of these experiments, and the funding support from the department. This is members of my lab, a few pictures from the last year or so. I'll be giving you a flavor of what we do from a number of these individuals up here today, while also acknowledging some of the work was done from some graduates, and we have our new cohort who just started a week or two ago. So this work's been funded regularly by ORSP mechanisms, as well as the Freshwater Collaborative of Wisconsin and the biology department. So, all right, so I mentioned zebrafish right off the bat, and so I like to address it square on. Why zebrafish? Why study this little critter that you can buy in the pet store? So this is a zebrafish on the right. This is not a zebrafish, but I like to show it because it's how I like to frame my initial thoughts about why the zebrafish is a useful tool to study questions of development and neurodevelopment. So yes, this is one of my daughters. She's a little bigger now, but I like to get to use this picture because I think all parents can relate to fun Halloween pictures. But the idea is that there is correlates, or at least, sorry, let me step back. They are not the same. Zebrafish are not the same as babies, but they provide some tools to study questions of related to the biology of babies and early human development. And we would like to think about it that way, to utilize the things that are similar in the fish to answer questions that are important about early human development. So kind of maybe more logistically, zebrafish are thought to lie on this continuum of complexity and throughput, and they can enable some answering questions about early development and biology that you can't do in humans, and they have specific features that make them suitable for questions that we're interested in. So one of them is that although the actual animal is different, at the cellular level there's a lot of similarities. So as I'll mention in a minute, this kind of idea about conserved molecular pathways drives a lot of why we think the fish is relevant to these kind of questions, because they do have the same kind of cellular and molecular signaling cascades and pathways going on that humans do. And they have similar responses to genetic and environmental factors, and can then be read out in some sort of phenotype as a living organism. And the other benefit for fish is it kind of is along the continuum of throughput and complexity, provides an intermediate to some of these questions. So on the one end, you have humans, which are super complex, but what we're really interested in, and if we could do it in humans, we should. On the other end, you have in vitro or synthetic assays at the level of DNA, or that's all in a test tube, and that has a lot of control over it, but is kind of far removed from the cellular context in humans. And the fish is somewhere in between. This is obviously written by a fish person because it fits them nicely right in the middle. But the idea that you can get some of the physiological complexity as a living organism and some of the higher throughput given their size and technical advantages. So what do I mean by that? One of the biggest things is they have this rapid early development. So I think the cool thing about working with fish is getting to watch them. It's one of my favorite parts. So I'm going to show you a little video it's actually from a publication, but we're going to look at the first 3.5 days of a fish's life in 90 seconds. So this is the scale over here in time. This is going from a single cell embryo, and you'll watch kind of how it grows. And remember, this is 3 days total. So I'll do a little bit of play-by-play on top to point out some important events. But first 4 to 6 hours, just a whole bunch of cell division. You've got a fertilized embryo. It's starting to grow. It's starting to make its capacity known. It starts to expand out, and you get in fish a process known as apibity, which wraps around the egg, or the yolk here, and you'll start to form kind of at 12 hours or so a body axis. So you can see kind of from here to this side. Oh, I slow it down if I put it there. So here's the eye and the head. Here's the tail end. And so this later part of the first day, it's a lot of growth, and you start to get somites and muscles, blood starting to flow. And about 1 day old, you notice kind of a curved fish-like creature. And the next day is a lot of it elongating out. You have, this is the heart beating away right there. You get the pigment cell development, and you get formation of the head and the jaw and the eyes up here. That's about 2 days. So already 2 days kind of stretched out. From here, it's mostly growing in size, although there's continued complexity up in the head, the ears, and the mouth, because it needs to be able to be forming the ability to go eat, because this yolk is going to last it for, it's getting smaller, about 4 or 5 days comfortably, and then it's going to want to go off and be able to feed. And it goes away. So all that happens in 2 to 3 days, which is just incredible. And so it's sort of an accelerated timeline to answer those kind of questions. And we take advantage of that in our studies. So if we want to ask questions about early vertebrate development, this is the system to do it in. Here's a few of the other kind of technical aspects that are helpful about fish. It is a whole animal and intact, so you're doing things in the actual context that they're occurring. It is one of the lower vertebrates, so it has some anatomical similarities to humans compared to other systems. When you're working with students, it often goes down to flies, worms, and fish, and this is one that can answer these kind of questions. So they externally fertilize and give you a whole bunch of embryos when they're cooperating, so you get lots of subjects and samples to work with, which is really good for students. Show you some images later, but they're translucent. You saw before those melanocytes came in that you can see everything, and we actually can manipulate them to keep them clear a little longer. They have the genetic similarities. So upwards 70-80% of genes are expressed that have a human homologue. And importantly, they're relatively cheap and easy to handle and fit under some special IACUC rules. This is a picture of our part of our colony up in Phillips, where we have a whole bunch of tanks with different kinds of fish. So they have some of these technical advantages and these biological features to ask these kind of questions about early development. So what can you measure in lab with fish? It's quite a bit. And these are a few of the things that we measure or are working on and then I'll share some examples from today. So you can measure organismal level development of tissues and organs. We do a lot of that with microscopy, taking pictures, what do these embryos look like. You can do functional outputs, so behavior, which we'll talk about in a bit. So it's kind of an exciting aspect to be able to do both cellular molecular work and behavioral outputs. And then molecular biology, doing manipulations and measurements at the cellular level of what's going on in these animals. So this is sort of a summary diagram of how I think about our projects, that we're taking a chemical or a genetic factor of interest, of motivation from a human disorder or some sort of purpose to study in the context of the fish, with a series of assays at different levels. And this is the even broader summary. Where do we get our inspiration from? We're often looking at either health-related or environmental-related interactions with early development, and then using the fish as a model or tool to investigate the impact of those on this context. So more logistically, what are we talking about? We have kind of an integrated approach where we have a series of types of experiments we can do and that students are working on that are usually trying to pull from a single source, one chemical or one gene or something like that, and look at it at different levels in terms of its effect on early development in the fish. So, and I'll talk a little about each of these. We mainly start here with measuring what does it look like. We do lots of titrations and morphology experiments, trying to define, as you probably guessed, most chemicals to do something poor to an embryo if there's enough of it. So we're trying to define where that level is and how that compares to human exposure or environmental regulation limits, if it hasn't already been done in the fish. And then measuring some functional outputs in terms of behavior in the intact system or gene expression as a cellular readout. And then I'll end with talking about how we are also adding on genetic gene-environment interactions with some gene editing. So one of the main areas in the lab is motivated for relating to neurodevelopmental disorders, so human conditions that seem to arise during this kind of time frame. And one of our main sources for that is autism spectrum disorders. So just a little background on that and why we think it's important to investigate this in terms of environmental factors. So early, or I shouldn't say early at this point, but for most of the time that we've studied autism, it was thought that it was mainly a genetic-based condition. So early twin studies and other genetic types of genome-wide association studies seem to indicate that it was highly inherited, or at least had high correlation. But kind of more the last decade or so, environmental measurements have improved a lot, and genetic experiments have gotten a lot larger to be able to get a more precise measurement of what kind of influence the genetics should have, and it actually seems to be less than we initially thought. Still pretty high, so this is already, about 10 years old of data, but more of in the 50-50 realm of like genetic inheritance versus non-genetic inheritance, which is then just dumped onto environment or experience. 50% is still very high relative to most human disorders, but it leaves a lot of room for environmental interactions as contributing to this. So the genetic research has been really important for establishing clues of how autism occurs at the molecular scale. So genes associated with autism tend to be related to neurodevelopment or synaptic plasticity or other types of kind of, placing it in this functional context. And one of those prominent areas or findings, regular findings, is the balance of excitation and inhibition in the brain. So there's excitatory neurons and inhibitory neurons, different functionally in the brain of either increasing or decreasing signaling. And regular findings associate changes in particularly inhibitory neurons, and then functionally an alteration in the balance of those two as altered in autism. So I'll come back to that because we are trying to test that in the context of how environment may be influencing autism. So less is known about the environmental contribution, frankly, in large part because we had a hard time measuring for a long time at a population scale or public health level of what people are being exposed to and how that would relate to health outcomes. But that has been an area of large growth, due in part to improved measurements of chemicals in the environment, as well as electronic medical records and other health correlates to get a sense of, well, there actually is a correlation here between the presence of this chemical or experience in the environment and health outcomes. So that's where, this is where we come in, or at least we want to be a part of these questions. And so the kind of basic question is, how does environmental factors associated with autism impact brain development? Do they? So if we get a correlation between a chemical in the environment and increased autism prevalence, does that matter at all? And if it does, how does it work? So, first we take these epidemiological findings and try to figure out how they can affect early development. Then we are also looking at, do they affect certain parts of the nervous system? One way to do that's different types of brain cells, excitatory neurons, inhibitory neurons, glial cells. And what are the functional outputs of any of these changes? And then kind of one of our other base hypotheses is that if these chemicals are relevant to autism, they may act on pathways that have been implicated or associated with autism as kind of an initial strategy to link cellular changes to this environmental correlate. So, all right. So I'm going to give you so this is kind of a higher level theory. I'm going to give you an example of what we're doing, and then we'll get into some data from lab related to these experiments. So one chemical that we're interested in, that we've been working on, is called methylene chloride. This is a volatile organic compound, and this is an example of the kind of data that comes out of epidemiology studies where they find an increased odds ratio for diagnosis of autism based on the presence of this chemical in the community. And that's all there is. It's just saying, okay, we measured this and we correlated it with health outcomes. So there seems to be, if there's more of this chemical around, there's a higher rate of diagnoses. But then we also often find this part, that there's no biological study of this chemical, and definitely not in the context of development. So that's where our group is trying to contribute and try to test that question of how do these things act in development, and are they relevant to autism? So I'll tell you about the little bit of experience we've been doing with this chemical. So volatile organic compound it's one, there's many different types of these, and they get interest in, they're actually very useful in an industrial sense. That's why they're around. Methylene chloride is used in a lot of industrial processes. The thing I can relate to most is the drinking bird toy. Does anyone know the drinking bird toy where you hold it and it evaporates and then it bobs? That's methylene chloride in there. So it's got properties for related to heat engines. So we do have, environmental regulations of exposure to these kind of chemicals, but a lot of the EPA regulations are mainly at the level of toxicity. So how much is, going to kill cells, or, but not in the sense of more subtle changes to neurodevelopment. So we're trying to fill in that gap. So what's this experiment look like? So we breed zebrafish and collect their embryos and then expose them to chemicals, right, usually about 6 hours post-fertilization. These are some early embryos. And then we're going to look at them across time and see how they change or respond to different levels. We tend to refresh the chemical regularly because it's kind of assumed to break down over time in water or evaporate of some kind. So approximately stable concentrations across the exposure. So this is an example of what this can look like in terms of data. So these are images, bright field images of larvae at various stages. And I'm going to point out kind of what we're doing with these is often just looking at what they look like first, and then noticing trends of different phenotypes, and then counting them. We're trying to classify based on the prevalence and severity of those changes. So some of these shown on the left. So over here is some edemas, both on the yolk or the heart. Spinal curvature, both vertically and horizontally, can often be found, as well as more underdeveloped or developmentally delayed and smaller eyes for methylene chloride, at least, have been showing up as regular phenotypes. And this is work of Carly Owens and Simon Gary. And we have been classifying them based on the prevalence of phenotypes. So there are lots of things that show up, and then it's trying to how we're going to categorize those based on on severity. So we make kind of rules of what we're going to count as present or not present, and then kind of score them typically on this kind of mild and major scale based on what they look like across the fish that are measured. If we do that, in these kind of experiments, this is some of the more recent data that we've been trying to determine what level of methylene chloride affects development. Shown here is percentage of phenotypes based on different treatments. So E3 is just embryo media. There's nothing in it. The vehicle has methylene chloride has to be emulsified in water, so it has to have an ethanol added to it to get it into solution. So this is really the vehicle treatment. And then these are 2 levels of methylene chloride in the parts per millions that Carly has been looking at. And the couple of things to note. So this is the percent of the fish that are showing certain phenotypes, either minor or major, shown in different colors here on the x-axis. The main thing that's trying to be shown is that there appears to be more phenotypes in these methylene chloride treated groups compared to the vehicle. And while the amount doesn't look that much bigger, it looks noticeably bigger, these are a pretty big sample size for the amount of fish that are being looked at. So we're still kind of quantifying this difference, but Carly's done additional titration experiments to see that below 200 actually looks more like these vehicle groups, and above 200 or 600 tends to be more phenotypes showing up. So we're thinking that the kind of lowest observable effect level is somewhere around 200, maybe slightly lower than that. And so that gives us a point to investigate for morphological features versus non-morphological phenotypes. So we're doing some similar titration type experiments with some other chemicals. So one that Simon's working on is molybdenum. Some of these other motivations are for environmental contaminants, not just human health. Molybdenum has been found, this is a map from southeast Wisconsin, having high levels of molybdenum in well water, and trying to figure out what that may or may not be an issue for. And then Celia and Ashley are working on methylmercury, which is a known neurotoxicant that I'll talk a little bit more later in the talk. So once we've looked at and defined kind of that there is an effect from its exposure, we are looking at other levels of effect on the fish. And so one of them is behavior and the functional output. So this is an example of we can do motor tracking assays where we have a camera that can watch the fish in a multi-well dish and see what its behavior looks like. So this is the realm of Nate and Nickel right now to assess changes due to factors on the behavioral output. And so what I think about it is, in part, broadly, that properly formed nervous system is required or important for typical behavior. So this could be a more subtle assay than a gross morphological defect. That's a pretty strong change. So we can detect movement changes due to variation and due to variables. And we've got kind of two examples here on the slide. So on the right, these are actually traces of those fish moving in the well with given different treatments. You can do acute manipulations like this, where we're just measuring under baseline or given an anesthetic, which slows them down and they don't move. Or you can manipulate the circuitry. Here's a removing inhibition on the system can lead to hyperactivity, shown in this blue area down here. Nate's also doing some induced changes, so I don't always want to give a chemical to change their behavior when I'm trying to see what another chemical is doing. So we have employed a kind of light dark assay because fish have a dark induced hyperlocomotion. So in this drawing here, or this graph here, we've got time on the x-axis in the instrument, velocity on the y-axis, and then 4-minute increments, we change the lighting. So the first 4 minutes, the fish is in the light, and then the light turns off, and the fish swims around more in the dark. And then the light comes back on, and the fish slows down again, and this can be repeated regularly. Here's just kind of two examples of this format. So this gives a way to make sure the fish can move, and then see if it can respond to this photo stimulus. So we're wanting to use this with our chemicals, but also have been establishing the assay to measure differences in the fish intrinsically. And this has been one of Nate's findings that is not entirely surprising, but is interesting and relevant for what we're doing. So we have a couple different strains of zebrafish, and we just wanted to compare first how they behaved. And it turns out they have different baselines. So we have the AB fish is the kind of standard genetic model, and the AK fish is a toxicology model. And so Nate went about kind of measuring them just to see how, see what it was like. We didn't expect them to be different, but lo and behold, they are different, kind of across the whole setup. So in both the light and the dark, the AB fish seem to move more. So what is stressed to us is it needs to be well controlled in comparing these kind of experiments, even at the level of which fish you use. So behavior is another area we can measure. And I'm going to pick it up a little bit to get through these last two ideas. So going scoping in, behavior scoped out, we scope in, we can do gene expression analysis. And so this is Jillian, Emily, and Cora are working on measuring gene expression across development in the zebrafish using quantitative PCR. So this is a technique that can be used to measure single genes in a relative fashion. So we can compare, is it more or less expressed given a treatment or a variable. So we have established this technique in lab to measure different neuronal cell markers. So this is where we're going towards that excitatory inhibition idea to measure those types of neurons and seeing if we can see changes at that level even when there's not changes at the morphological level. Because frankly, the morphological level changes is pretty high, and we're interested in more if there is functional changes, if the fish looks normal or typical, than if there's a big morphological change. So, my students have been actively getting this together, and this is one example of what the data can look like. So we're measuring, up here are measurements of RNA that's been converted into DNA for the PCR reaction, and the dye signal on the y-axis. As you cycle through PCR more, you get more copies, and then that binds more dye, and you get a signal that arises. Down in the bottom is using all the same primers, but just not converting the RNA to the cDNA molecule that's necessary. And this is proving the specificity of these reagents to measure the genes of interest. So all of these curves up here are shifted or way to the right, or they don't exist in this bottom sample, suggesting that you need to have the RNA version of the gene to be measured. And so we are measuring kind of various neuronal marker genes that are only expressed in a certain kind of neuron and using that as an output. So now that we have these reagents, we're establishing the baseline expression. When do these genes turn on across normal development, so that we can then compare them to a chemical or genetic manipulation and see if they change those time courses, which we interpret as changing how those cells develop. All right, I'm going to mention briefly this last project, and I will have to leave some of my slides for time, but I've talked mainly about environmental factors. We're also interested in how genetics will play a role or interact with those environmental factors. So we've been establishing the capacity to make gene editing in the lab to create fish that have knockouts of genes of interest. So this project with Celia and Ashley has been creating zebrafish lines with genes that are involved in methylmercury metabolism and knocking them out to then look at human variant effects in development. And this is a collaboration with the Carvan Lab at Milwaukee and funded by the Freshwater Collaborative. So this is using CRISPR-Cas9 technology, where we inject Cas9 protein and a guide RNA that targets a gene of interest in a one-cell embryo. And it is a nuclease, a targeted nuclease activity. So it creates a mutation that we hope, or we will try to find fish that have null alleles or non-functional versions of the gene. And then we're using what's called high-resolution melt analysis, which is a variation on the qPCR, to genotype these fish and see if we can detect differences at the target mutation. So just a couple examples. This is some earlier experiments to test all of this using a pigment gene, because part of the whole reason for genotyping is the fish doesn't necessarily have a visible phenotype. So this was using our experimental approach to target a pigment gene. You can see the fish over here often have lighter pigment or lacking pigment cells, especially you can look at the eyes for a lot of these, they're clear. So that's evidence that this pigment gene was mutated. And over here is the corresponding HRMA data, where the fish on the right are shown graphed in blue and are different from the fish that are on the left. These are two types of controls. and we would expect those to be about the same on these graphs, so the green and red being close together. So having that in hand, we went ahead to try to mutate some genes that we don't know what the fish will look like. And here's some of our data from that, where the control fish or sequences are shown in red and yellow, and they are similar in terms of this curve. And the fish injected with the CRISPR reagents are shown in green. And it's similar to the blue is actually a synthetic sample that simulates a 10 base pair deletion. So we were trying to see what it would look like in this assay if we had a mutation, what we would expect it to the data to look like. And the main message here is that the green lines are closer or more like the blue lines and distinct, importantly, from the red and yellow. So we've collected these fish down here that have this signal and are raising them to then try to breed to see if they passed on these mutations and try to characterize what those specific mutations are. So, all right, I'm just at the end. I wanted to put a plug in for ORSP and involvement with undergraduates. I know everyone here needs no introduction to working with undergraduates, but this is Celia and Ashley presenting this work recently at Society of Toxicology and the Research in the Rotunda event. I'd really commend the Research in the Rotunda event to any faculty to take your students. This was a really great experience, shown over here in the Capitol building in March. And on the slides, I put the links to some of their video abstracts that were produced with IMC and are just really, I get excited watching our students talk about their research and be excited. So I hope you can enjoy that as well. And this is where I want to end, back kind of overview of what we're doing. So I've given you some examples of how we're trying to assess environmental influence on brain formation, and where we get the ideas from and how we think about it, and some specifics of the techniques and approaches that we're using. I'm always open to talking about it and looking for collaborators or people who are interested in these kind of questions. All these students will be presenting at Circa, so if you're interested in hearing more, I'd really encourage you to stop by their posters and presentations. And thank you. I'll be happy to take questions, and thank you for your time. Thanks.
Yeah. I love the fact that your work is being inspired by epidemiological and public health kind of, human studies that then you're looking for an appropriate model. And on one of the slides, we were showing the epidemiologic data. Unless I missed it, it looked like all but one of those epidemiologic studies, the confidence interval included the null value. And there was one study that showed a statistically significant difference. So if you were starting as the base with that set of epidemiologic data, what sort of inspired you to go beyond the statistical lack of correlation, because you clearly showed effects in your animal model, that. So what made you look beyond that set of data and explore this?
Yeah, so that's a really keen observation. So the questions about the epidemiological inspiration or connection to the autism prevalence data, and the fact that the confidence interval overlapped the null, and it I'm just repeating for those remote. Why choose that one or go in that direction? And it's a good question. So a couple points on, so the epidemiology data for these type of associations does tend to be kind of not very strong at this point. And some of it's due to that sampling issue of trying to measure these chemicals and associate them with the diagnoses kind of data. why we chose methylene chloride in this particular example was kind of twofold. One was the consistency of the findings of being in a certain direction and a certain level of odds ratio. So it wasn't, statistically always, 2.0 or more or some sort of, amount, but it was always in the same direction and had a direct correlation with autism. There is more of this chemical, more autism. And the other thing was more biological, that there was very little known about this chemical in terms of early development. So so part of me is hedging, wanting to learn something about a chemical that may be relevant to autism, but is potentially interesting and not much known about it anyways, to then characterize it. So we kind of go back and forth in lab about whether to do chemicals that we don't know anything about, because then we have nothing to compare to, but we think they might be important, or something that's more established to then be able to link into the data or the literature more. So in the end of the day, I don't know if this will have a direct link to autism, but we felt there was enough of interest of the chemical and its wide use, that it would be worth understanding more about. So, you're welcome. It's a really good question. It's something we're always going for, about whether the physiological relevance, the clinical relevance, and what are we going to be able to learn or not learn from this kind of approach. We are not going to necessarily solve the autism association. Our approach is mainly going to establish whether there's a biological consequence to this chemical in development.
Well, thank you, Dr. Carter. I think if we have time, we can take the discussion offline, but thank you very much for the presentation. And please join us next week, which will be the last staff faculty forum for the semester. Same place, same time. Thank you.
Thanks so much.